The NAP4DIVE will generate two new tools to test nanoparticles that are generated to deliver drugs to the brain over the blood-brain barrier to treat diseases such as neurodegenerative diseases and brain cancer. The project is funded by the European Commission. The two new tools are a design simulator and a blood-brain barrier on a chip. The design simulator will screen thousands of different nanoparticles designs and suggest the best designs that will be then tested in the blood-brain barrier on chip. The accuracy and the physiological relevance of these tools will be tested by our industrial partner. We are a multidisciplinary team of 11 partners from 6 different European countries, and the project is coordinated by Åbo Akademi University in Finland. The team consists of biomaterial scientists, IT engineers, technology developers, biologists, biophysicists, experts in ethics and health economics. The NAP4DIVE approach aligns with the incentive of the European Union and the world to reduce animal testing in biomedical research and drug development. We collaborate with external bodies, including standardization bodies such as JRC, neurosurgeons and policymakers to demonstrate market readiness and promote the use of non-animal models in the biomedical industry.
We will produce 5 types of synthetic nanoparticle and 1 type of biological-based nanoparticle, and do different kind of surface modification for screening their capability to deliver drug across blood-brain barrier. The nanoparticles will also be utilized as model nanoparticles for BBB chip design and also for feeding data for AI modeling. The promising nanoparticles with good drug loading capacity, biocompatibility, BBB permeability, and in vivo stability will be proceeded further for large-scale manufacturing. The protocols for synthesizing, characterization, and quality control will be documented.
NAP4DIVE in-silico model comprises of 3 different components. First, a nanoparticle design library. Second, a simulation environment that models the brain-blood barrier structure and its interaction with nanoparticles. And finally, a machine learning component that predicts different properties of any given nanoparticle design without the need to run lengthy and costly molecular dynamic simulations. To build our library, we start by retrieving the data from public repositories, publications, and any open database. Along the course of NAP4DIVE project, we enrich our data with our own experimental data and any new findings. The simulator based on machine learning models is able to predict the efficiency and functionality of different nanoparticle designs. We later test their validity and their functionality in vitro on our brain-blood barrier and chip models.
We will create a microfluidic blood-brain barrier on chip tailored for the needs of the pharmaceutical industry. High throughput, cost-effective, compatible with standard instrumentation, and mimicking the blood-brain barrier physiology. Our system outperforms existing options, including cost savings and faster manufacturing times. The most promising nanoparticles recommended by the design simulator will be tested in vitro in the microfluidic chip.
AstraZeneca will be an end user of the tools delivered through NAP4DIVE. We will test a nanoparticle initially successful, but where that success was due to a compromised blood-brain barrier. We will also look at nanoparticles who have failed in clinical trials to see if NAP4DIVE tools could have predicted that failure without the use of preclinical animal models. For the validation purposes, we will test NAP4DIVE nanoparticles against the current gold standard, which is animal models, to test for efficacy, pharmacokinetics, and accuracy. We at all times will be in strict adherence of the 3R principles for running animal studies in the context of NAP4DIVE.
NAP4DIVE will create new ethical guidelines for the development of organ-on-a-chip and AI technologies in the pharmaceutical industry. The idea is to bridge pharmaceutical realities with animal ethics debates from philosophy and ethics. We will develop an ethical framework for contributing to the trustworthiness of the blood-brain barrier on a chip and digital nanoparticle simulator in the NAP4DIVE project. This will be done by having a dialogue between different stakeholders to get their input about creating scenarios for what could happen, just after the project with these technologies, and then linking it back with a decision tree to outcomes and decisions that are made in the project.